Welcome to our screencast, the grapefruit juice case. Granny believes that eating grapefruit is good for digestion because it's acidic. She thinks that the acid in grapefruit helps the enzymes in the liver to function. To test her theory, we conducted an experiment using a reaction chamber, liver enzymes, reactants, hydrogen peroxide and catalase, and a graduated cylinder to measure the products of the reaction. We placed a paper disc soaked in enzyme and 20 milliliters of hydrogen peroxide, separate on opposing walls inside of the reaction chamber, before closing it with a rubber stopper. The reaction chamber was connected to a tube with its other end inside a graduated cylinder, filled with water, so we could measure the gases produced. We then rotated the reaction chamber to bring the two parts into contact, and we observed the amount of gases that was produced.
In this experiment, we changed the amount of acid or base in the 20 milliliters of reactants. We had 10 milliliters of hydrogen peroxide, which was constant, and the other 10 changed to have our 5 test cases. Our test cases were all hydrochloric acid, 5 milliliters hydrochloric acid, and 5 milliliters water, all water, 5 milliliters sodium hydroxide and 5 milliliters water, and all sodium hydroxide. We hypothesized that as the pH of the reactants deviates from 7, the efficiency and activity of the catalase will decrease. Therefore, grapefruit juice, which has an approximate pH of 3 to 3.3, will not help the function of the liver enzymes.
Next, we will be looking at our lab's observations, which are displayed in this data table, where oxygen gas is measured in centimeters and has displaced the water in the test tube over 30-second intervals in our 5 solutions of 100% acid, 50% acid, 50% water, 100% water, 50% base, 50% water, and 100% base. The water group with a pH of approximately 7 has the greatest amount of O2 displacing the water in the tube. The acidic groups yielded the least oxygen gas, where the double asterisk indicates 1 bubble of O2 released. The first trial of 100% base is an outlier to the experiment and did not match our previous data, nor research, and was far beyond both. The results were then entered to a scatter plot, where the trend lines indicate a moving average. As time begins to reach the end, the rate of reaction and O2 release begins to slow. The circle trial indicates an outlier of 100% base group. This next graph shows the two 100% base group variations side by side, with the blue as an outlier group. This is a graph of expected enzyme activity in an optimum pH of 7. However, since our experiment yielded far more O2 for the base trials, our graph would look more like this.
At a molecular level, the hydrogen peroxide substrate approaches the catalase enzyme, and as they bind, the bonds in the substrate are weakened. Here's a molecular view of this process, and eventually two products are formed, in this case water and oxygen gas. You can see the oxygen gas in the form of a bubble here, surrounded by water of course, and that gives us our net equation of 2 hydrogen peroxide molecules creating an oxygen gas molecule and 2 water molecules, of course being catalyzed by the catalase enzyme. And our experimental results were congruent with our hypothesis in the sense that the pH level at which the beef liver catalase was most active and led to the most rapid rate of reaction was equal to the pH at which the liver typically operates, that being 7. However, when the pH environment was changed above or below 7, the rate of the reaction decreased. This is because hydrogen ions in acidic solutions and hydroxide ions in basic solutions are charged and therefore interfere with the hydrogen and ionic bonds that hold together the enzyme, because they're going to be attracted or repelled by the charges created by those bonds. This interference causes a change in the shape of the enzyme, and specifically its active site, which is no longer as accommodating to the hydrogen peroxide substrate.
One of the improvements that can be implemented is to create 14 different solutions covering all the pHs of the scale. By testing smaller increments, a curve of the rates of reaction versus the pH of the solution could be graphed, allowing us to make more specific conclusions about the relationship between the environmental pH and enzymatic activity. Exact concentrations for solutions can be produced from stoichiometric calculations, as opposed to using our eyes with litmus paper. The inconsistent amount of catalase which was collected on paper discs would lead to inaccuracies. The beef catalase was collected with a paper disc with no form of measurement. Improvement would be to have capsules, which would stick to the glass bottle. Fermentation is a process involving enzymes where a compound rich in energy is broken down into simpler substances. It is also identified as a process where large organic molecules are broken down into simpler molecules by microorganisms working anaerobically. Yeast undergoes fermentation, and the results of this lab suggest that it would be more effective to measure the optimal pH range of the enzyme and maintain that during the course of fermentation to increase its efficiency in industrial situations. The question that this experiment raises is whether there is variance in the optimal pH environment for different enzymes. The data provided from testing the optimum pHs of different enzymes would either confirm or deny the hypothesis that all enzymes have different optimal pH ranges. In conclusion, Elizabeth's grandmother was incorrect because our results show that the catalase's optimum pH is 7.